Hi all, I'm Poorni from the Department of Biotechnology. I'm handling the subject called Bioprocess Engineering, and I'm going to take the topic offline and online method for a biomass estimation and flow injection analysis. So what do you mean by the biomass estimation? First we will see. So how this biomass estimation is going to be produced in the case of your fermentation process. So when we are making the organisms to grow in a particular media, the organism will utilize those nutrients for their growth and for their reproduction. Why it has to be growth? It has to be growth in either in the cell number or in the cell mass, so that it will be helpful for producing the product which might contain your desired as well as undesired. So by utilizing all the nutrients from the whatever the raw materials we have been provided, the microorganisms utilizes those raw materials and it produces a product in the case of a stationary phase, so that it will be easy to derive the particular product, but there will be a disadvantage of an undesired product also will be present along with the desired one. So how we are going to estimate the cell biomass and how we are going to derive those products that we will be seeing. So when we talk about cell growth, the cell growth is said to be, it is based upon both your replication and the change in the cell size. So it undergoes a physical, a chemical, and a nutritional condition, so that it utilizes those nutrients, and based on that the microbial biomass has been increased with respect to the time. So which can be this as how this nutrient has been utilized or adapted by the microorganism, which means that when we are adding the cells to the substrate, the cell starts to utilize the substrate so that it is getting converted into the product. Either the product might be the intracellular product or an extracellular product. So along with your this product that will be formed in your biomass. So it will be containing a more number of cells as the cell is being able to reproduce the product along with the cell. So when we talk about the rate of a growth, how it is being directly related. So here the rate of a growth is directly related to the concentration and how the cell is getting reproduced. So how the cell is getting replicated once it is utilizing the nutrients which are present in the media, which will be uptake by those nutrients, so that it is able to reproduce or increase in both the cell concentration as well as in a cellular reproduction, that is based on a number of cells. So based on this, we can give the rate of a microbial growth at a particular time. It can be represented as a nu net is equal to 1 by x into dx by dt. So here when we talk about a dx by dt, it is nothing but the x represents the biomass that has been changed with respect to that of the time by based on the utilization of the nutrients by the microorganisms for their growth in a particular time period. Okay, so here when we talk about a net specific growth rate, this specific growth rate can be related with the gross specific growth rate, in which can be represented by the equation called your nu net is equal to nu g minus of kd. Always the nu net unit will be of a time inverse. It might be the hour or second or a minute. So when we talk about a nu net, that is equivalent to your nu g minus of kd. So substituting those equation in the case of your nu net is equal to 1 by x dx by dt. So nu g minus of kd is equal to 1 by x to dx by dt it comes. So when your exogenous metabolism is being taken place, your kd will be equivalent to 0. So what happens? The nu net will become directly equivalent to your nu g. So whatever the gross growth rate is there, that will be acting as a net specific growth rate of a microorganism. So when we are inoculating the organisms in a particular media for their growth, how it might be either a batch or fed batch or a semi-continuous or any form of your continuous. So how it is being cultured. So here when we talk about your microorganism is being cultured in a vessel where your cell is there, in which it will be utilizing those nutrient media for their growth. That can be done both in the industrial scale as well as in the laboratory scale. So when it has been added, it will be started to utilize those nutrients which are being supplied as a raw materials in the medium, so that it starts to get adapted, so that it will be turning that into the particular product. So that product can be of a purified form. It consists of either a desired as well as a undesired product. So then how we can quantify the cell concentration? So we can quantify the cell concentration in two methods. The first one is the direct method. The next one is an indirect method. So whenever we talk about a direct method, the name itself indicates it is a feasible one due to the presence of a compound that has been present in the medium for estimating the cell mass concentration. In which how we can quantify the cell mass, it can be quantified for both the direct and indirect method in the terms of your cell mass or in a cell number. Okay, so always the cell mass concentration is preferred to be the measurement for your cell number density. So here instead of a cell number, because it will be replicating fast, so that instead of counting the number of a cell that has been taken for your growth estimation, we can measure the biomass, that is the cell mass that has been formed during at a particular time. So this is your phase, as we all know that this is your growth phase that has been plotted between the time on an x-axis and number of cells on the y-axis. So here when I talk about your first phase, which is being explained as a lag phase, that is said to be the first initial phase, where your microorganism starts to adapt the nutrients that is being supplied in the case of a nutrient medium. So once it has been started adapted, once it is fully adapted, it is entered into the next phase, that is said to be the log or an exponential phase. So when you talk about the log or exponential phase, here the number of cells which will be starts to divide, which means that it will be able to reproduce into a number of cells. So next it will entering into the next phase called a stationary phase, which means that it is said to be taking the source. Once the number of cells has been entirely divided, it is ready for your product formation. So that happens occur in the case of a stationary phases. So once in the stationary phase, the product has been formed. This product, which will be comprising of both your desired as well as an undesired as a byproduct, then it starts to decline in that is the final stage of your any growth cycle if you take for a microorganism of a bacteria. So here in the death phase we talk about that will be a nutrient depletion, since all the nutrients has been initially utilized by this in the case of this lag and the log phase. So here in the case of a death phase, as your product has been entirely formed in the case of the stationary phase, the nutrient depletion has been occurred, so that the product will be ready to extracted or isolated from the stationary phase in the form of your biomass. So how this biomass can be measured? So here in this case of a measurement of a microbial gate, this can be done based on a different method. The first one is a dry cell mass or a wet cell mass, absorbance based on a cell count. That's we are doing it manually under the microscope, or if it is of a computerized, how we can do the counting the cells and those whether the cell is viable or a non-viable. That is a viable cell count, that is a live cell. So when we talk about the method of measuring the microbial growth, these are based on a different parameters like your dry weight and a wet weight or absorbance, the same thing that has been. Apart from this, it can be utilized for measuring the cell mass concentration in the form of your nephrometer and turbidometer and your spectrophotometer. Everything can be used for a estimation of your cell biomass. So when you talk about a wet measurement, how we can measure? So it is an easy step. We will be making the organisms to grow in a particular media, so that once it has been grown by adapting all the nutrients and once started to form the product, this sample of a cell which will be collected in the centrifuge tube. Once it has been centrifuged at a particular RPM for a certain period of a time, then the supernatant that has to be removed, which consists of an unwanted debris, cell debris, which will be present in the case of a supernatant. The down part will be having the pellet. So that contains your biomass. So that contains your desired and an undesired product. So we are after your centrifugation, we are going to drain the supernatant. We are going to weigh the centrifuge tube along with the pellet. And before starting the centrifugation, we should measure the weight of your epandref tube, that is empty one. So the wet weight minus of the empty epandref weight will give you the wet weight measurement. That is how much amount of a cell biomass has been measured. Okay, so the dry weight measurement, this is similar to your wet weight. Like we'll be collecting the sample, centrifuging the sample, draining of the supernatant so that the pellet has to be weighed off. But here, when you talk about, we will not be measuring the weight of a pellet in a wet form. So we are going to make it air dry so that we'll be weighing the centrifuge tubes once it has been dried. So that we are having the term called a dry weight measurement. So similar to your wet weight, we have to measure the empty centrifuge tube, then the dry weight as a pellet centrifuge tube weight minus of a empty centrifuge tube will give you the dry weight measurement. Then the third one is the absorbance. So here for your batch growth, for this one, how we can measure the number of cells based on the absorbance value. So based on a time, so for at a particular period of a time, that is for a 0th hour, 1st hour, 2 hour, 3 hour, the particular period of a time, we are going to measure the absorbance value. So that absorbance value, when we are plotting, it will give you the phase like this. So that's why we call this as a growth phase of a bacterial cell. So this absorbance, it can be done using your spectrophotometer or a UV visible or UV spectrophotometer or a colorimeter, any form of a absorbance value. So OD value will be taken on and plotted against the x-axis so that it gives you the absorbance value, which means that after plotting, it will give you the curve like that. That is called as a growth cycle. Okay. So the next one, how this cell number of a cell can be measured, that is of a manually, okay, or like your online. So here we talk about how the cell number can be measured. The cell number can be measured by placing the microorganisms here. One loop full or a pipette full of a culture will be taken and it will be placing in the case of your here. Once it has been placed, we'll be keeping this cover slip so that the culture what we have been kept, that will be smudged here. So this we are going to take under your microscope and this will show the how many number of cells which will be present. So this is said to be the hemocytometer, or we can call this slide as a Petrov-Hauser slide, since it has been discovered by Petrov-Hauser to detect the viable as well as the non-viable cells. So the main disadvantage is it will be able to see the non-viable cells also. Okay, so that's why it interrupts the process, so that we will be going to the next method. Okay, so here when you talk about your this thing, this is said to be the Petrov-Hauser slide or a hemocytometer, and when we are visualizing this slide under your microscope, you'll be getting a number of grid lines like this. So here, how many cells which will be present in each and the corner of your grid lines, we are going to measure the number of your cells. So once this is an empty hemocytometer for your counting of a squares. So here in this case, if we keep a microorganism over here, once it has been covered with the cover slip, the microorganisms will be smudged. Right? So once it has been smudged, how it is going to give the number of cells, that absorbance can be done like this one under your microscope. Okay, so this is your Petrov-Hauser slide, which we will be having your, once if we are pouring your microbial culture here, then if you are visualizing, it will be showing both your viable as well as a non-viable cells. So each square, how many number of cells that has to be counted, and those cells for each cell, how many cells is there, how many number of your organism is there, microbial cell is there, that into the 25 will give you into 50 into 100 will give you the number of cells. So always the number of cells in the case of your hemocytometer is said to be the manual counting rather than your automatic online method. Okay, so here in this case, when you talk about, it gives you the main disadvantage is said to be your viable cells can be not only the viable cells can be detected, non-viable cells also will be detected, which means that the dead cells also will be taken into the count. So this has some disadvantage because of that. So that's how this will be having a square of a 20. So 20 into 20 into 50 mm. That will give you how many number of cells in this case. It will give you the average number of cells can be calculated based on the number of bacterial original sample have been taken. Either it's a 1 ml or a 100 ml or a 50 ml or a 250 ml that has to be counted off. Okay. So the second one is the colony count. So here it's nothing but we will be taking the bacterial grown culture, we will be plating it on an agar plate, and once it has been solidified, we making it to cool after your sterilization. Then we'll be placing the bacterial cell culture over on it and spread it on a plate using your L rod method. Okay, so using your L rod, sterile rod, if you are doing an L type of a streaking, then it starts to form a colony around this. This can be visualized by staining so that it gives you the number of cells in a colony forming unit. That's why we call it as a CFU. That is a colony forming unit. The next one is the third one, how we can measure of our online method, that is a particle counter. So here in the case of a particle counter, if you talk about, it is used for measuring the cell number as well as the cell size distribution. So what happens, we will be placing the solution, that is an electrolyte solution, in a chamber. Inside the chamber we will be placing your tube, that is an orifice tube. So inside this tube we are going to place your electrolyte 1, and when we talk about your electrolyte 2, that is the electrode that has to be kept inside your electrolyte solution. So when we pass an ammeter, when we pass a current by applying the voltage, so here the cells that has been present in the case of an electrolyte, that will be secreted or pass through the force, and it will be touching your electrode so that it takes your signal in and displayed in a displayer in the form of a height of a pulse. That height of a pulse will give you the number of cells. So the height of the pulse, that gives or directly proportional to the number of cells which are present in your electrolyte solution. So I repeat here, this commercial particle counter here in this case, there will be a chamber in which your orifice tube which will be located. Inside to this orifice tube will be plating your electrode 1, so that the another electrode 1 which will be placed inside your electrolyte solution. Once we are passing the current, once we are applying the vacuum, the passage of a current through the voltage which will be making the cells from the electrolyte to be passed touch the electrode, so that it will be taking your output in the form of a height of a pulse as a display. Okay, so that directly gives you the number of cells. The next method is the turbidity method. So when you talk about the turbidity method, it is a simple and a rapid method for the growth estimation. But here the turbid, which means that more number of cells will be occurring, so that it will interrupt your processes. Okay, so why, in the sense, if the media has been a more turbid once the product has been formed, that making the light to be passed through the suspension in a limited amount. So it doesn't make a approximate, it doesn't make your accurate method for a cell growth measurement, but this can be done also for your cell growth measurement. So it gives only the approximate value rather than giving an accurate form. So these are the measurements for your microbial growth. The first one is a direct count, that is a counting chamber, that's a colony counting chamber. Electronic counter is setting by the flow cytometer, which means that a particle counter, right? So the next one is a viable counting method, that is a spread plate and the technique, spread and pour plate technique, that is said to be a colony counting, colonies forming unit that can be measured, and most probable number, that is the number of cells of a bacteria, can be measured. So the next one is your indirect method. So when you talk about indirect method, we will be doing it manually. Instead of doing it in a online method, we'll be doing it in a manually. So here the dry weight analysis, wet weight analysis, and the turbidity, which comes under your measurement of a cell mass by the indirect method. Okay. So the direct counting, as we all see, we have seen about a bit of a slide for your cells, for your counting of a viable and a non-viable cells, colony counter for your CFU by your spread plate method. Okay, so here then how we can measure the viable count. So what do you mean by the viable? The viable is said to be the live cells, right? So the viable cell is defined as a cell which is able to divide and increase in the cell numbers. Right. So how we can measure in this case? It can be done using your serial dilution. So you'll be taking a 1 ml of your sample that will be taken as a parental one, and from there we'll take an 1 ml and pour into the next one and making to an 10 ml, which will be making a 10 to the power of 1 dilution. Then it will be taken to the next. From here we'll be taking a 1 ml and we'll be making it to 9 ml. Why? So similarly we will do for a n number of your tubes, so that it gives you the alone only the viable cell count. Okay, so it will not disturb you, the non-viable cells will not disturb you in this viable cell count. So it will be used to give only the viable cell. It's for screening and isolation of a specific microorganisms for the product. The last one is the spread count method. So this is a set with the spread count method that will be deriving your cell mass, so that plating on an agar plate and spreading on it with the L rod, and the plate is incubated overnight so that it makes you the colonies. The colonies visible can be done by the staining technique, so that it gives you the colony forming units. So based on a number of colonies formed, it can be measured. So these are the various methods that merits and demerits. So when you talk about the microscopic count, it is application in the case of your all your laboratory conditions. So, but what is the disadvantage in the case of a microscopic count? It cannot able to distinguish your viable and a non-viable cells. Okay, that's why cannot be able to distinguish the live cells and the non-living cells, because it will comprises both your live cells and the dead cells. So the next one is a viable cell count. Why we go for a colony counter in the sense it gives only the viable cells. So it is very sensitive if your plating is said to be not in a sterile or an aseptic condition. So we have to make the plate to be in an optimal condition. Then when you talk about turbidity, the turbidity, if the product of your microorganism that has been produced due to it is more turbid, there's a possibility of a light is very least, so that it is said to be cannot be able to determine the cell density. So that's why this is one disadvantage, but this method is a fast and a non-destructive one. So the next topic we'll see about a flow injection analysis. So when we talk about a flow injection analysis, what do you mean by the flow injection analysis? So we are analyzing the sample through some flow. We are going to inject the sample so that it is able to detect what type of a chemical which is present in it. Okay, so we are taking the cell biomass for your estimation whether the product or desired or undesired has been formed. That can be flow through your analysis technique for analysis is said to be the flow injection analysis. So when you talk about a flow injection analysis, it is said to be the first generation of your flow injection analysis. So that is why we say it is a first generation of a FIA for analyzing the chemical. It is used to analyze the chemical of your any analysis that can be done. Either it might be a metal or iron or atoms or any form can be done. So here, which means that how this analysis can be done, how the sample is being taken, and what are the supporting one that takes a sample for a analysis will be seen. So here in the simplest form, your red zone is said to be your sample, which is being carried by the reagent or a catalyst that we call it as in a carrier, that has been denoted here as a blue form. Right. So once the carrier has been taken the sample, this will be pumped through the next reservoir for your mixing up of the carrier as well as the sample for a analysis. So here in the next region we'll be placing the detector. Once the mixing has been done properly, it will be taken to the detector for the analysis, so that it gives you the display in a display form, a digital value, what are the chemicals that is being present in it. So that's why we have said it is the first generation of FIA technique, and it is most commonly used for analyzing the chemical. Here the sample zone is being taken to with the or mixing up with the carrier in the mixing tank, and which will be moving downstream so that the sample solution gets dispersed while the product is being formed, which will be taken to your detector that records a change in the color or another parameter, and that recorder or of a detector that gives you the display value in the form of a digital value through the flow cell. So how this instrumentation or a basic setup is made up of? So here when you talk about the instrumentation or a basic setup, here in this case we will be having the carrier, right? So this carrier, it has to be mixed with the sample. So how this carrier is taken or mixing up with the sample, in the sense we'll be making the sample to be get into your mixing coil. There is a mixing tank, and for your mixing up with the reagent or a carrier through the pump. So mostly common pump used in the case of a FIA analysis is the peristaltic pump. So it will be injecting the carrier for your reaction with the sample at a particular period of a time gap. It will not be making it to be in a continuous flow of a carrier for a sample injection, for a carrier injection, for the mixing up with the sample for the detection of your chemicals, what is present in this case. Okay, so here it has been mixed up in this case of a mixing tank, then it will be taken to the detector level. So once it has been detected, it will be taking your base, it will not be recycled back to the FIA. So it has to be treated as the base, and it will be connected to the display here so that it displays the value what of the chemical analysis has been done. Okay, so it is said to be the simple and manually operated system, which is comprised of your peristaltic pump, mixing tank reservoir, the walls of for your sample, as well as your carrier for mixing up in the case of a mixing reactor. And this mix, once it has been done, it has been used to detect the level of a chemical what is being present, that will be displayed in a display. Okay, so here when you talk about FA, here it will be having the light source, which is said to be the tungsten light source, and as well as your spectrophotometer, that is a training tool for a sample analysis. So that is a basic step or an instrumentation for your FIA. So when you talk about a FIA, there are three categories it falls up, four categories it falls up. The first one is a FI, that is a flow injection. The next one is a sequential injection. The third one is a bead injection. The last one is said to be with the purification technique, the automated nowadays feasible and a advanced form of a technique also it has come, that is said to be the sequential injection chromatography. So once a product has been analyzed for the particular chemical analysis, this will be taken for your purification based on a color or based on a molecular weight. For separating the product in a particular molecule using a chromatography technique, that is being coupled with the sequential injection, which is said to be the flow injection analysis coupled with the chromatography. So when you talk about FI, so we have said the flow injection is said to be one directional, that is said to be the unidirectional flowing carrier stream to which your sample are going to be added. So here, once it has been taken to the reagent, to the mixing zone, the concentration difference will be occurred that form due to the dispersion. So this dispersion has been taken into the four category. Okay, that I will be explaining it later. As it passes through the detector, it is used to detect the chemical analysis, and once it has been detected, the base will be taken for the draining out, whereas the detector value which will be taken to your recorder, which will be displaying the digital value so that it will be recorded for our analysis of a sample. The next category is said to be the sequential injection. So here in this case, the flow of a carrier and a reagent, which will not be in a unidirectional one, it is said to be a reversible or a bidirectional one. Okay, so here it is used to optimize the protocol so that it will not be affecting the entire process for analyzing the particular chemical analysis. So when we talk about your SIC, that is said to be the sequential injection chromatography, which is being coupled with the chromatography technique that uses a programmable flow of to separate the components based on the difference on your chemicals. When we talk about a flow, continuous flow method, so the types there are various types in which the carrier is taken to the for the reaction with the reagents. So when we talk about the types of your flow method, the first one is your continuous mixing method. The first one is a continuous mixing method. So here the sample is injected into the system using your carrier in which it will be taken up at a particular period of a time continuously. The mixing will be taken place for your analysis to be done in the case of your waste. So that can be done. So which means that this after passing through the detector, once the display or digital value has been analyzed, it will be recorded in a display screen, so or displayed in a display screen and sending the waste to be drained off, or it can be recycled back. Okay, so the next method is your flow is said to be the stopped flow continuous mixing method. So here in this part, it is used to prevent the air from entering into the system so that the systems will be operating in a continuous manner without stopping of a flow, and either in the case of a reagent washing also. So the flow is stopped between these two operations. So during your system is getting mixed and when the reagent is washed out, which means that once a waste has been drained off, the continuous flowing method should be stopped off for a certain period of a time so that it can be used for a analysis of chemicals in the case of a detector. Then there is said to be the third is a continuous flow titration. It is a large dispersion technique, which means that it is used to, instead of a mixing coil, it should be making the dispersion process, that is the diffusion process to be occurred so that the reagent or a carrier will be mixed up and then the color will be detected. So here when you talk about the principle, the sample that's said to be the analyte. What we need to analyze, this will be injected into the carrier solution so that it can be mixed up through the dispersion technique. So the dispersion technique, which means that it might be either a radial or a conventional diffusion with a regular period of a time will be mixing up, right? So here when we talk about your diffusion mechanism, the main principle which behind is the dispersion coefficient. So when the sample after your mixing, it will be passed to the detector so that for a analysis of a detection of a sample, it will be turned into the waste which can be recycled or it will be drained off as a waste. Okay, so the next one is the decrease of a sample in the reagent consumption and a base generation. So sample, it has to be analyzed in a smaller amount for a accurate measurement, and the reagent should be consumed in a lesser amount and the waste should not be generated, or if it has been generated, it has to be recycled back. So this is the basic setup. So here we are going to inject the sample. Where we are going to inject the sample? When the reagent or a carrier is being taken from the reservoir to the profiling or a mixing unit, here we are going to add the sample, which will be mixed up here and it will be detected by the detector. Then it will be drained off as a waste and the result will be displayed or recorded in a display form. How? So here in this case, what are the various steps for preparing your setup or the instrumentation of an FIA? So the first is your sampling. So when we talk about a sampling, the sample is measured, that is said to be the analyte that is making or injected into the carrier stream for a detection of your chemical analysis. Okay, what type of a chemical? So this can be done with the help of a sample injection board. Then the sample processing. So the second stage is the sample processing. Here we'll be taking the analyte that will be taken through the dispersion or a diffusion technique into the detector so that the detector can be done. So here, once the detection, so the third stage is your detection. Once the detection has been done, the waste will be drained, generated, that has to be drained off, or it can be regenerated back into the FIA, but without the interrupting the process that has to be done. If not, it has to be drained off entirely, and those direction recording the values so that it can be used to record the chemical analysis what it has been present in the case of your analyte. So that is a detection. So the next one is your how this diffusion has been occurred. So this diffusion can be occurred in a two forms. The one is a radial form and the next one is a longitudinal diffusion. So here we said there are two types of diffusion that has been occurred based on the dispersion. The first one is the radial diffusion. Next one is your longitudinal diffusion of a fluid. So as I said, it is based entirely, the diffusion mechanism is based entirely on your dispersion coefficient. So which means that whether the analyte is passing in the form of a radial diffusion or it is passing in the form of a longitudinal diffusion. So here the dispersion coefficient can be used as a measurement term which is defined as a ratio of the concentration of your sample before the analyte, that is before getting into the mixing, divided by the after mixing up, so the peak concentration at the detector level. So here the concentration of the sample at the time of the entrance in the detector. So the D is said to be the dispersion coefficient, which is defined as a ratio of the concentration of the sample before and after the dispersion process has been taken place. So before the mixing and after the mixing, those ratios can be set as a dispersion coefficient, that is the capital D. And this C0 is said to be the concentration of your liquid in the injected volume, and capital C is said to be the concentration in the case of a detector once it has been mixed up. Okay, so here the diffusion process, how it has been occurred, it is based on a dispersion of your analyte that has been taken place into a different forms of a species for detecting what type of a chemical species is present, or in the form of an atoms it is being present. So this dispersion coefficient, what are the factors that affects the dispersion coefficient? So your sample volume, your tube length of your carrier, how it is, what length it is, and what is the flow rate, and those ID. Okay. So when we talk about a dispersion process, there are also four types of a dispersion process, which is used to detect your dispersion coefficient. So the first one is an injection, the second one is a dispersion, third one is a detection, and fourth one is a washout. So here, when we talk about an injection, so this can be explained with this diagram. So here, when we are making the carrier to be passed from your reservoir, here the sample is getting mixed up with the carrier, so that it is getting mixed up in the case of your injection, then it is taken for your detector level. Okay, so once the carrier is there, that is mixing up with the sample. Here the sample is getting in. So here the sample is getting mixed up with the carrier. That is means that the dispersion process has been occurred, which means that the diffusion is being occurred. So that the sample, we call it as an analyte, that is converted into the species. That is into the n number of an atoms. So here this once the n number of atoms has been segregated, this will be used to detect the sample. That is an analyte. That's why we have a term called as detection here. Then once it has been detected, so this value which will be given as a peak value, which is used to determine what type of a chemical which is being present in our sample. Once it has been deducted the analysis, the waste which will be can be washed out or it can be recycled back for your FIA analysis. Okay. So here when we talk about a FIA analysis, this analysis always done in a manual method, but nowadays as the technology has been increased and the availability or need of the requirement for your chemical analysis, this has been an automated flow injection analysis has been done. So that system we call it as a modern flow injection analysis system. This usually comprises of a basic setup, what we have seen in the case of a manual, the injection wall, the pump, your reactor, where the reaction is being carried out, the detector, and the anode sampler. So this can be used for your any number of a chemical analysis in the case of your large scale industries or chemical or unit process or any process. Okay. So here we'll see what are the various walls we do have. So when you talk about an injection wall, we are going to pass the sample here. So when we need, the loop will be opened up here so that the sample will be injected through that way. So if not, the loop will be closed. So this syringe will be made of this wall, which will be loading the sample, which will be having a closed condition. Whenever we need the sample, that is the analyte, to be reacted with the carrier, the loop will be open so that the sample will be mixed up to your mixer tank for the mixing of with the carrier. Okay, so here this gives you the high precision, in the sense it gives you the high accuracy value, and it is of an automated one. It doesn't need that operator to be monitored, as it is of a modern flow injection analysis. So what type of a pump we use? As I have said, that we use a peristaltic pump, that if the flow rate can be controlled for a particular period of a time. So after a particular period of a time, we are going to give your carrier, or it is going to take your sample of some amount into the mixing stream for a mixing up. Okay. So this flow rate has to be designed in such a way for taking the carrier to be mixed up with the reagent. So what type of a detector we use? So the detector we use, it's of a UV visible or IR, it might be or ICP-OS or ICP-AES or ICP-MS-AES, or a GC to be coupled with the FIA for a N number of your folds. So when you talk about a detector, we use a UV visible spectrophotography, and most recently we are using your IR spectrophotometer for analyzing the atoms or molecules that has to be coupled with the FIA. So once a chemical analysis has been done, how this product can be purified? So this product can be purified either the three techniques. The one is the dialysis, then the extraction method, then your gaseous diffusion, so that the liquid can be evaporated, so that it will be making the solid to be in the form of a product that can be made. So the separation in of FIA is done by either a dialysis. So we have to pour the sample in a dialysis membrane and keep it for a dialysis overnight, so that the protein or whatever the sample we need as a product in a desired form, that will be separated out or purified out. And also it can be used your liquid extraction, and also by your gaseous diffusion.